Hello, we're glad you've joined us for this live webinar, Applications of Targeted NGS Panels for Clinical Research of Primary Immunodeficiency Diseases. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Thermo Fisher Scientific. Thermo Fisher is a world leader in serving science, whose mission is to enable customers to make the world healthier, cleaner, and safer. The clinical sequencing division of Thermo Fisher Scientific offers solutions for next-generation sequencing under the Ion Torrent brand of products, which include Ion AmpliSeq panels, Ion S5 sequencers, and Ion Reporter software. For more information, please visit www.thermoscientific.com/Ion Torrent. Let's get started here. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can pose questions to the speakers during the presentation while they're fresh in your mind. To do so, simply type them in the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or you can report your problem by clicking on the green Q&A button at lower left. I now present today's speakers, Janet Chou, MD, and Wayne Bainter. Dr. Chou is an assistant professor of pediatrics at Harvard Medical School and an allergy immunologist at Boston Children's Hospital. Her research focuses on the genetic and molecular mechanisms underlying human primary immunodeficiencies, with an emphasis on identifying rational targeted approaches for immunomodulation. Dr. Chou's experience as a clinical immunologist drives her search for more precise and rapid ways of understanding immune dysregulation. She received her MD from Harvard Medical School and completed a residency in pediatrics and a fellowship in allergy and immunology, both at Boston Children's Hospital. Wayne Bainter is a research technologist at Boston Children's Hospital. His expertise encompasses the application of genetic approaches in the study of human immune disorders, functional analysis of the immune system, and characterization of murine models of human disease. In his current role, Wayne is leading the Targeted Next Generation Sequencing Initiative in the JEHA laboratory at Boston Children's Hospital. He graduated from Salve Regina University with a BS in biology and a minor in chemistry. Dr. Chou will now begin her presentation.
Thank you, Judy. So today I'm going to discuss applications of targeted next-generation DNA sequencing to primary immunodeficiency research. As most of you know, primary immunodeficiencies are genetic diseases that impair the immune system, resulting in breaches of host immunity that normally is responsible for immune surveillance and self-tolerance. As a result, individuals with primary immunodeficiencies develop severe or life-threatening infections, autoimmunity, and/or malignancy. Primary immunodeficiencies occur globally, although they are rare diseases. They are most common in areas with high rates of consanguinity due to founder effects and the predominance of autosomal recessive primary immunodeficiencies. However, these diseases do occur through all modes of inheritance and are found in all populations globally. So we study these diseases truly to identify novel mechanisms of host immunity because knowledge of genetic etiologies underlying primary immunodeficiencies has defined and leveraged cancer research tools, the development of immunomodulatory agents, and studies of inflammation and self-tolerance. So in going about this research, there are three main strategic challenges. The first is genetic diversity, and there are approximately 10,000 genes expressed in the immune system. And of these, about 300 monogenic causes of primary immunodeficiencies are known, with about 10 to 15 new genetic causes identified annually. So it's a rapidly moving field. The second strategic challenge is the phenotypic variability, in that one disease may have many different phenotypes, or conversely, deleterious variants in different genes can result in similar phenotypes. So the genotype-phenotype correlations can be difficult to predict. And thirdly, there is a level of technical complexity because the biologic assays of the immune system are essential for characterizing these disorders. But these assays are highly specialized. They require viable cells from study participants, and they are costly due to reagent expertise that's necessary to conduct them. So I'm going to first give you an overview about how we approach the study of these disorders and how we identify these mechanisms, and then I'll delve into applications in our own research. And I have these three main approaches categorized almost historically. The first being educated guesses, which is the longest standing approach, where one uses the knowledge of immunologic and signaling pathways to predict and define what a new primary immunodeficiency might be caused by. So the advantages of this approach is that primarily it builds logically on existing foundations of knowledge and methodology, but it's very challenging to identify entirely novel pathways using this approach because, of course, it's built on what's already known. The second approach that's widely used now are similarities to mouse models of disease. So the main advantage of this approach is that the mouse models show us the in vivo relevance of candidate genes and the effects of various variants. However, as we all know, mice are not men, and so there's differences between the murine and human phenotypes, and it may be due to the species differences or the environmental differences or even epigenetic differences. And thirdly, the most new and perhaps the most widely used now are the unbiased genetic approaches for identifying mechanisms of PIDs. So the main advantage of this approach is that one can discover truly novel causes of PIDs using this unbiased approach. The disadvantage is that there is a lot of data that's generated, which is certainly manageable, but more pertinently, these approaches give very little information about the true biologic effect of the variant. So the way we conduct our research is through an international consortium for immune deficiency, which is spearheaded by Raif Jaha, who's the chief of the division at Boston Children's in immunology. And it incorporates a network of collaborators from 19 countries in Europe, the Middle East, Northern Africa, North and South America. So it's really a global collaboration. And we have an annual study subject enrollment of over 150 individuals per year with immunodeficiency phenotypes. And the hub of this consortium is at Boston Children's, where we have a group of 6 people with collective expertise in basic and clinical immunology, genomics, and molecular and cellular biology. And I would say that this makes up part of our work, but not wholly. And so in our aim to define novel genetic and molecular mechanisms underlying primary immunodeficiencies, speed and throughput are really key factors in how we do our research. So the repertoire of tools that we use encompass 3 main groups, which basically fall into those 3 approaches that I previously discussed. The first involves in vivo models, where you start with the study subject phenotypic data, and one may close and recapitulate the human disease by making a knock-in or knockout mouse model of that phenotype once the causative variant is known. The second main group of tools are, I call them functional assays or biologic assays, and broadly speaking, they can be classified in terms of studies that look at protein expression, cellular functioning, and molecular signaling. Obviously, these are extremely broad, and within each category, there's a plethora of specific techniques. And thirdly are the genomic technologies, which include microarrays, Sanger sequencing, targeted next-gen sequencing, and whole exome and genome sequencing. So in categorizing these tools, you can see that the in vivo models and the functional assays share the strengths and the weaknesses. The strengths being that they are essential for the characterization and understanding the mechanisms underlying these disorders. But the weakness is that it will not identify the specific genetic variant that underlies this disease. On the other hand, genomic technologies complement the other 2 approaches in the sense that the genomic approach will identify the causative variant, but provides no or very little information about the biologic effects of the variants. So for this reason, we incorporate all 3 approaches and all of these tools in our search for mechanisms that underlie primary immunodeficiencies. So I'm going to show you how we have done our research both in the past, which did not involve targeted next-gen sequencing, and in the present, which does. So previously, we would have recruitment of study subjects with PID phenotypes. And for each study subject, there would be at least a 2 to 4 week period of phenotypic investigation. This includes functional assays, Sanger sequencing of candidate genes. And if there was no candidate gene that was identified, we would then outsource the whole exome or whole genome sequencing. And that would take at least 2 to 3 months, sometimes longer, depending on how complex the analysis was. And if we were able to find a candidate variant, we would proceed then onwards to the mechanistic investigation. So this is really the work that proves the biologic effect of each candidate variant. And we often will recapitulate the human disease in a mouse model. And you can see the length of time is really quite longer for the mechanistic investigation, lasting somewhere up to 3 or sometimes longer years. Using this approach, we sequenced over 100 study subjects using whole exome or whole genome. And our yield of variants that we found to be causative of the phenotype was about 55%. So this is greater than what's been published in some of the large, more recent studies in the New England Journal and in JAMA about the use of whole exome sequencing in finding genetic variants. Those studies published a percentage of about 25% yield. And I would say our increased yield is partly due to the fact that we focus on a specific disease type, that is immunodeficiencies, of course. And also we're able to incorporate more than just genetic assays. So the phenotypes and the functional assays are important in really characterizing us and letting us understand the phenotype very thoroughly before we start analyzing the whole exome or whole genome data. So the lessons we learned from that approach, actually, was that the majority of subjects had an identified etiology, which was really just an atypical phenotype due to a deleterious mutation in a gene that was already known to be associated with immunodeficiency. So all these study subjects were screened by senior immunologists, and even with that initial screening process, we had unexpected phenotypes. And we found that the personal or family history of life-threatening infections was the only variable that was significantly associated with identification of a causative variant, and that other factors such as consanguinity, gender, age of symptom onset, and age at the time of the study enrollment did not increase the odds of identifying the causative variants. So we found that interesting because it showed us that this approach could be used beyond the consanguineous populations and could really be used in a more global approach. But that also showed us that because most of the variants were found in known genes, that we were spending a lot of our resources and our time finding things that were already known, and we needed a better and a faster throughput method. The strength of this approach found that it's mechanistically detailed, and we did have proven ability to identify novel etiologies of primary immunodeficiencies. About 10% of the study subjects had a novel cause of disease that had not been previously described in any of the medical literature. The drawbacks, as I mentioned, are that it was time-consuming and the cost. So based on this, we really wanted to find an approach that would let us focus the bulk of our resources on new biology and new mechanisms. So with that aim, we incorporated a targeted next-gen sequencing approach using the INS5 system. So you can see that the main difference in this approach here, highlighted in blue, is that after study recruitment, all subjects undergo genetic screening using this targeted next-gen panel. The panel we use is called the primary immunodeficiency panel that's provided by Thermo Fisher. And my co-speaker, Bainter, will go into the technical details of the panel use and the metrics and the data output. But in terms of the big picture, we basically have three outcomes. Either after screening with the panel, we identify a clear etiology, a clear causative variant, and the study is complete. No more resources spent on a known gene. Then there's also possible variants. There may be several of them. And so then we will then move on to mechanistic studies to determine which, if any, of those causative variants would be associated with the phenotype. And finally, the most interesting, of course, would be those that have no known etiology. That is, there were no variants that were found on the pre-screening. And those study subjects undergo whole exome and whole genome and mechanistic studies. So you can see that by triaging the study subjects using the targeted panel, that we're able to focus more of our resources on the cases that have unknown etiologies. And I will show you just an overview of the outcomes and how that's increased our speed. So using this approach for the past 1.5 months, we have actually found an increased yield of causative variants. So using this approach, we've had a yield of about 80% compared to 55%, which we had previously using our initial whole exome approach. Part of this may be due to the fact that we're able to achieve higher coverage at lower cost using a targeted panel. And it is much easier using this targeted panel to identify large exonic deletions than combing through the whole exome copy number variation data. Our throughput has increased tremendously by 7-fold. So we're able to process at least 70 samples per month, and will probably increase as time goes on, versus about 10 samples per month using our old method. With this higher throughput and with the targeted panel, there's a lower cost for samples with mutations in known PID genes. That's because the cost of the panel itself is far cheaper and far less time-consuming than doing all of the functional assays and the Sanger sequencing that we had done manually previously. So this really enables us to focus our resources on research samples with potentially novel mechanisms of primary immunodeficiency. In terms of logistics, it's much easier because DNA is the starting material, and there's a very small amount, only 10 nanograms, that's needed. And certainly this is much easier to send internationally than viable cells. And from our experience, as Wayne will detail, it's quite simple to establish this in-house compared to setting up whole exome sequencing in-house. So in terms of the drawbacks, there is a larger upfront cost for the S5 system, the Ion Chef, of course, than outsourcing the exomes on a per-sample basis. But cumulatively, the cost per sample will be less using the new approach that incorporates targeted sequencing. It's just that any technology will require an initial outlay of funds. There is a short training and learning curve that we will discuss. And then finally, the last drawback is not specific just to targeted next-gen sequencing, but really to all genomic studies in the sense that they will never replace mechanistic studies for functional characterization or for whole exome sequencing for identification of novel genetic causes of immune deficiency. So I'm going to present just 2 short case studies. And the purpose of these case studies is really to show you some real metrics about the differences in our old approach versus the new approach. So the first study subject is a 17-year-old woman with a history of intermittent neutropenia and common variable immunodeficiency, which is the most common immunodeficiency worldwide. It is largely attributed to defects in the humoral system, so immunoglobulin secretion and B-cell function. But the specific genetic causes of CVID are very few compared to the number of individuals who do have this disorder. This particular study subject had a history of sepsis, disseminated intravascular coagulation, and necrotizing fasciitis, and unfortunately, ultimately died from an inflammatory episode thought to be macrophage activation syndrome. So it was a very severe phenotype. Using our old approach, using a functional approach, we would have done flow cytometry for TB, NK, memory B cells, autoreactive B cells, B cell proliferations, and in vitro immunoglobulin secretion from B cells. And that's just the starting panel. And that would have taken, as you can see, about 3 weeks of work. And from a genetic perspective, as I mentioned, the majority of individuals with common variable immunodeficiency have no identified genetic etiology, although there are at least 12 known genetic causes of CVID-like phenotypes. So Sanger sequencing all of the exons for all those possible genes, which would obviously have been even more time-consuming and costly. So running this study subject through the NGS panel, we identified a homozygous 7 base pair deletion in the gene CECR1, which was a frame shift leading to a stop. This gene encodes ADA2, which catalyzes the deamination of adenosine and 2'-deoxyadenosine to inosine and deoxyinosine. And it's released by macrophages, monocytes, and dendritic cells. So ADA2 has been described, but largely it's been known to result in early onset stroke and vasculopathy in most, although not all, cases. And combing through the literature, a small percentage, about 15% or less, of the reports of this already rare disease have hypogammaglobulinemia and decreased memory B cells. So although the mechanism driving this defective B cell function are not known, it's increasingly becoming realized that this disorder has a humoral component that really resembles CVID. And then zebrafish models have shown also that this enzyme is important for neutrophil development and maintenance. So the challenging aspects of this case are that it really is an atypical features of ADA2 deficiency. The study subject had no vasculopathy or early onset stroke, and the predominant feature was one that's found in only a minority of patients, which is CVID. But you can tell that based on the severity of the mutation, that is a frame shift and a stop, that this would be the most likely genetic cause of the disorder. And the other thing is that since this patient had passed away, there wasn't any further clinical research material from her, and so the study wouldn't have been able to be done using a functional approach even. So what this was able to show us is that we were able to find an atypical phenotype of what is a known disease, but also to divert, as I mentioned before, more of our resources to new biology. And then the second case I'm going to show you is also another case of common variable immunodeficiency in a 12-year-old male with a history of refractory pneumonias and bronchiectasis. He had a history of recent interstitial pneumonias due to possible viral etiology, so that suggests that perhaps there might be a defect in his T-cell function. He also had pancytopenia suggestive of myelodysplastic syndrome and had monosomy 7 that was found, and was able to receive the BCG or the TB vaccine, which is a live vaccine without any complications, which in contrast to his lung disease suggests that maybe he does in fact have intact T-cell function. So the difficulty of this case is that the phenotype is not well defined. It's hard to tell whether this is truly a CVID-like or whether it's more of a combined immunodeficiency with defective T-cell function. So again, using our old functional approach, we would have had done flow cytometry, proliferation, immunoglobulin secretion, and lymphocyte proliferation, all of which would have taken, again, over 3 weeks. And the candidate list when the phenotype is undefined like this is vast, so it's virtually impossible to pick genes to start Sanger sequencing. And so running this patient through the next-gen panel, we were able to find a homozygous missense mutation in ADA. So this is different from the previous ADA. This is an isoenzyme, so this gene encodes adenosine deaminase 1 instead of 2. And this disorder is known to have a lot of phenotypic variability. Classically, it does present a severe combined immunodeficiency, which is the most severe immune deficiency, with an absence of B, T, and NK cells in early infancy. Although a small proportion, about 15%, will have later onset at 6 to 24 months of age. And there are rare cases of individuals with this from 4 years of age to adulthood in terms of their onset of presentation. This particular mutation has actually been previously reported to be disease-causing, and it has been associated with delayed onset of symptoms in older years. So again, this shows very atypical features because this individual had a very mild presentation and was able to tolerate a live vaccine that typically patients with a combined immunodeficiency would not be able to have without any symptoms. So these two case studies show two putative distinct genetic etiologies of CVID, and they show how using the targeted panel really expedites the work compared to the original functional or Sanger sequencing-based approach in terms of identifying mechanisms of primary immunodeficiency. And so in conclusion, I'd just like to emphasize, of course, that PIDs are very heterogeneous, and that's what makes the field both interesting and challenging, and that there's so many genetic causes, and that they really require a multidisciplinary approach that incorporates genomic, biologic, and in vivo models. And that this approach may be helpful in discovering new mechanisms of host immunity in both non-consanguineous as well as inbred populations. And finally, the lesson that we really learned in changing our workflow was that the targeted sequencing approach significantly increased our throughput and decreased the cost per sample, which permitted us to have a very sharp focus of our resources on novel mechanisms and biology. And so I'd like to thank, of course, the group at the Division of Immunology that's spearheaded by Raif Jahab. And you can see the members of the core consortium team, Wayne Bainter, Michelle Massad, Craig Platt, and Brittany Kengemi, as well as the collaborators that referred to these specific cases to us. And then finally, all of our collaborators who are part of the consortium. Thank you.
Thank you, Dr. Chou, for your presentation. A poll question will appear on your screen. You may select an answer and then close the poll question by clicking on the X in the right corner. And the question is, are you interested in adopting the ION AmpliSeq Primary Immune Deficiency Research Panel for your research and testing needs? And when the poll concludes, I will then turn the presentation over to Wayne Bainter. I think the poll has now concluded. So Wayne, whenever you're ready for your presentation, please begin.
Okay, well, hello, and thank you all for joining us today. As Judy said, my name is Wayne Bainter, and I am a research technologist at Boston Children's Hospital in the Division of Immunology. To complement Janet's great talk on the applications of targeted sequencing to the field of primary immunodeficiencies, I'm going to present the highlights and practical aspects of the ION S5 system. Before we purchased this system, we compared it to other options and had many questions about the real-life workflow, hands-on procedures, and data analysis that goes into the system. My talk today will address all of these questions and topics. I know many of you are familiar with NGS, but for those of you that are new to the world of NGS, I will be giving you an overview of these steps before delving into our out-of-the-box experience with the ION S5 system. Additionally, since I'm going to discuss the granular aspects of our data, I will also give a synopsis of the technology used by this system. Our setup utilizes the ION Chef for automated library preparation, template preparation, and chip loading. This instrument significantly reduces the amount of hands-on time and variances that occur with manual library preparation. Once we have a loaded chip, it is put onto the ION S5, which then performs the sequencing and subsequently aligns the data and also performs variant calling. In this overview of the sequencing process, you see that the first step in the sequencing process begins with library preparation on the ION Chef. We begin with 8 samples of DNA that undergo a targeted multiplex PCR known as an AmpliSeq reaction by Thermo Fisher. This then amplifies the 264 genes on the Thermo Fisher PID V2 panel. During this multiplex process, each sample is ligated with an adapter and also a unique barcode, which then allows for each library fragment to be recognized by the sequencer as a specific sample. Once amplification is complete, the libraries are purified, pooled, and equalized at a concentration of 100 picomolar. Now that we have a pooled library, we're ready to begin template preparation, which also takes place on the ION Chef. Template preparation begins by binding each library fragment onto a bead using the adapter added during library preparation. Once bound, each fragment undergoes clonal amplification via an emulsion PCR. This then fills the bead with one unique library fragment. The full bead is also known as an ion sphere particle, or ISP, and then this is then loaded onto an ion semiconductor chip, which contains billions of microwells that each ISP sits in. A chip containing full ISPs of all library fragments is then loaded into the ion S5 sequencer. The ion S5 uses unique semiconductor technology which utilizes the change in pH during nucleotide incorporation to detect the library sequence. The S5 begins the sequencing process by flowing single nucleotide solutions over the chip. When a DNTP is incorporated to the leading unpaired template nucleotide, a hydrogen ion is released and the pH within the microwell is then decreased. That change in pH is then detected and recorded by the instrument. After the flow of the first DNTP, each well is washed out and a new flow with a different DNTP is started. This process happens for each of the 4 DNTPs for a total of 500 flows. For the ion S5 system, Thermo Fisher offers 3 different semiconductor chips to accommodate different coverage needs. Each chip has different capacities for read length, ranging from anywhere from 3 to 5 million reads on the ion 520, all the way to 60 to 80 million reads on the ion 540. The right chip depends on the number of amplicons in your panel, the number of samples, and the desired sequencing coverage that you need. For the PID V2 panel, we're aiming for at least 95% of all of our bases with a minimum coverage of 20x. The PID V2 panel has approximately 5,241 amplicons. For a real-life example, we found that running either 8 samples on one 530 chip or 32 samples on one ion 540 chip will enable us to achieve at least 20x coverage on more than 95% of our bases. And we found that sequencing more samples on a larger chip is definitely more cost-effective. However, the smaller chip is very useful for small runs that need to be performed urgently. Now, one of the big questions that we had before committing to the ion system was the actual hands-on time required to run the instrument. Now, this chart here depicts the time required to set up each step in the ion system. As you can see, the bulk of the hands-on time is actually consumed by DNA isolation and preparation. And the ion system requires 40 minutes of hands-on time for only 16 samples. In total, each sample requires approximately 51 minutes of pre-analysis time, of which about 45 minutes is actually just DNA isolation. You can see that the use of the Ion Chef enables a rapid load-and-go approach with minimal time for library preparation. Another important concern is subsequently the hands-off time needed to run these instruments. Here I show the time it takes to run 8 samples on one 530 chip. As you can see, the total hands-off time is approximately 35 hours, which enables us to do other work while sequencing is going on. For our workflow, I set up library prep first thing in the morning on the Ion Chef. About 8 hours later, I remove the sample and load and prep the machine for template preparation, which then will run overnight. The following morning, I will then load a chip into the Ion S5, and it will be done sequencing in about 3 hours. And at that point, the data will then be uploaded onto the cloud, and analysis will then take place. We found that this approach enables a rapid and convenient workflow. I'm now going to discuss the more granular aspects of our data and our experience with references to our results using the Ion S5 system. When we first purchased this system, we did not have any experience with in-house next-generation sequencing. We had a 3-day training with a field application specialist and a bioinformatician, and then that following week we were fully operational and ran our first chip without any issues. I'm the one that's responsible for the sample preparation, machine operation, and data analysis, and the data is then cross-checked with my colleagues consisting of physician scientists and senior scientists. In just 1 month, we've already studied over 70 samples, and which, as Janet mentioned, is 7 times faster than our previous approach, which included functional assays and Sanger sequencing. Now, I just want to give you a quick idea of the run reports and the data that's generated by the Ion server once sequencing is complete. The run report is a summary of run quality metrics that go on in the chip. Now, here's an actual heat map of one of the chips we ran. The heat map indicates ISP density and chip loading percentage, thus giving feedback on how well the chip was loaded. This is a very important QC check because it can indicate if any mechanical issues with the chip occurred during loading. If the chip is unevenly or incompletely loaded, the coverage of the samples will be lower than desired, and you can see that clearly on the heat map. The run report also generates ISP metric statistics, and then this shows us some of the most important information from our sequencing run. You can see that the total reads of the run at the top, which for this chip was over 20 million reads. This number will vary depending on the size of the chip used, and this is on the high end for a 530 chip, just about 20 million reads. Another important statistic shown is the clonality of the library fragments. This will tell us how pure the ISP reads are because the goal is to have one library fragment per bead. Now, a run with low clonality or high polyclonality indicates that the beads have more than one library fragment per bead library. And we found that the clonality of approximately 60% generates good data and complete data. Lastly, I want to highlight the final library percentage. This tells us what percentage of reads were low quality. As you can see from this run, 9% of the chip was low quality, and then it can also show you the test fragments and the dimers that occurred during the run. And then this final slide of the run report shows the average read length. And we expect this to be around 200 base pairs while having very few samples below that 200 base pair mark. And as you can see, this run was spot on with an average read length of about 208 base pairs. Now, once sequencing data has been aligned to the reference sequence, it is then uploaded from the ION server to the ION reporter. Now, this software is easy to use and annotates our variants. The ION reporter will then generate an Excel spreadsheet, which can then be easily filtered and further annotated for our use. And as you can see here, it will generate an Excel spreadsheet that will include the chromosome number and position, the different types of mutations, the minor allelic frequency, the location of the variant, like whether it's an exonic or splice site or intronic mutation, the gene name, the codon sequence, the exon number, amino acid change, polyphen and CYP scores, which are very important to us because they predict the effect of the amino acid change and help us determine if it's pathogenic or not, as well as the reference ranges to other databases, which is also very useful. Now, for our own data, we use very few factors to filter out all the sequencing data. First, I filter out using the minor allelic frequency, or the MAF, for all the variants which are less than 1% in the population. And this is because of the rarity of primary immunodeficiencies in the human population. We then focus on the exonic and splice site mutations and eliminate all synonymous mutations from our initial analysis to prioritize those variants which are most likely to be deleterious. And finally, I filter based on the polyphen and CYP score, as I mentioned, because this helps us determine the pathogenicity of the variant. Now, this chart here compares our in-house generated data with data generated by Thermo Fisher for the PGM and Proton. Now, Thermo Fisher generated this performance data for the PGM and Proton using the PID V2 panel, which we also use. And I've compared that, and I've compared my results from 5 different 530 chips to these runs. And as you can see, our data from the S5 system is definitely comparable to the other predecessor instruments. However, you can see that there's the coverage at 100x is lower than both machines. Now, one thing to keep in mind is that these sequencing runs on the ION S5 were performed with 8 samples per chip, as opposed to the 2 or 6 samples on the PGM or Proton. Now, if we were just to load 2 or 6 samples like the PGM and Proton, we would definitely have more coverage. And another way to increase the coverage would be to run these 8 samples on a 540 chip, and this would allow for far greater reads per run. And one thing to keep in mind, too, is that we did not aim to maximize the number of bases having a 100x coverage. And this is because our research focuses on germline rather than somatic variants. In our experience so far, we've had some minor technical and mechanical issues. But what has been more important to us is not the issues itself, but more the resolutions to these problems. And the response and expertise from Thermo Fisher's NGS group has been absolutely superb and has been superior to any experience we've had so far in the lab. We've had real-time support both in person and over the phone, Their staff has been leading us to complete resolutions to all the questions and issues that we've had. And in closing, I just want to emphasize 3 aspects of our experience. And this is the setup and operation of this system was fully functional after just 3 days of training, which is pretty amazing. And we've had excellent results so far, which has tremendously increased our pace of our research, as Janet mentioned. And what I think sets Thermo Fisher and the ION S5 system apart is the simplicity, speed, and support offered by this whole group. And in closing, I'd just like to thank Dr. Raif Jahad, the PI and the Chief of the Division of Immunology at Boston Children's Hospital, Dr. Janet Chou, my supervisor, mentor, and partner in science. And also I'd like to thank Dr. Michelle Masad, Brittany Kangemi, and Dr. Craig Platt for their assistance with sample collection and coordination. Thank you very much.
Thank you both for your presentations. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Chou and Wayne Bainter will answer as many questions as time permits. Oh, and we have a second poll question for you here. And so the question is, are you interested in getting a quote for the ION S5 sequencer? And so if you will take a moment to answer this question before we jump into the Q&A, that would be great. And while you are working on that, I will pose the first question. And the first question is, how often do you find 2 mutations simultaneously for primary immune deficiency in next-generation sequencing? And how do you decide which one is a causative mutation?
So I think that's a great question because primary immunodeficiencies are classically considered monogenic diseases. That is, one mutation, one gene, one disease. But what we are finding is that increasingly there are modifier mutations or diseases that can be caused by 2 simultaneous mutations, as this question poses. In our experience, in terms of what I will consider 2 distinct pathogenic variants that lead to the disease phenotype, is definitely in the minority. I would say it's probably, in our experience, about 5% or less of the study subjects that we have found. But there are a few other modifier mutations, such as those in the FMS gene that causes familial Mediterranean fever gene that is more common in the population in the Middle East. And that I wouldn't consider as much of a pathogenic, it's more of a modifier, can accentuate the phenotype. And so that might be higher, maybe closer to 10%, if you include those kinds of lesser modifier genes. Now, how to sort out which mutation is which causing which disease or its contributions really can be only done on a functional approach. So we either try to dissect the 2 pathways by stimulating them in ways where only one pathway and one mutation will be evaluated versus the other. The other alternative approach is to generate cell lines, each having only one mutation or the other. And or one could do it in a very expensive way with a mouse model. And the third approach is actually to correct each mutation individually and or together, either using lentiviral transduction or transient transfection. So as I had mentioned before, the mechanistic part of it really has to be done through a functional approach. But the targeted and the whole exome approaches are what's needed to find all of these mutations that are dual in nature, because each and every one of them has been a surprise to us. We've actually never predicted a dual primary immunodeficiency, and we would never have found it without a genomic approach.
And our next question is for Wayne. How are you managing your sample information and downstream data? Are you using a laboratory information management system?
So we're managing it just on our on the hospital server, and we're actually, one of the best parts about this ION system is we're making use of the ION cloud that is provided to us. So this has really helped us organize and keep our data straight.
Okay, the next one is for Janet. Dr. Chou, will you be updating the panel to include more genes as more genes are discovered that cause primary immune deficiency?
Yeah, so that's definitely something that we are going to do. The way we decided to do is to start first with the panel and see how that worked out. And because it's worked so well, we'll be updating probably annually. And in our discussions with Thermo Fisher, our understanding is that the adding of the custom genes actually was quite doable and not hard at all. So given the pace of the field and the fact that 10 to 15 new genetic causes, I would say, are identified each year, we'll do an annual update and then combine them in pools with the existing panel.
Thank you. The next one is for Wayne. How many samples do you typically run on each of the S5 chips, and is it more cost-effective? One second. More cost-effective to just batch and run samples on the 540?
Yes. So usually I try to run 32 samples on the Ion 540 chip, and as you can imagine, that takes about a week to do all the library preparations for that. So I try to do 32 on a 540, and that is definitely more cost-effective. But in some cases, some samples are more urgent, so I can run just 8 samples on a 530. But if you're going for, down to the last penny, definitely running 32 samples on a 540.
Is there a presence of T cells in Zika-affected patients? And if so, is it possible, one second, let's jump here, and is it possible to have enough to counteract the apoptosis, that is, degeneration of bone and brain tissue in infants with microcephaly, with necessary adaptations such as scaffold building, self-assembly, and polymerization in regenerative biology?
Sorry, can you repeat the question from just in the first part? I didn't hear the first part. I'm sorry.
Sure. Is there a presence of T cells in Zika-affected patients? And if so, is it possible?
Right. So it seems to me that this question is asking actually about, so everybody has T cells, of course, and the infection with any virus such as Zika or even the common cold or HIV stimulates both the innate as well as the adaptive immune system. And so I think this question is more one that's looking at mechanisms of host immunity, and perhaps asking why some people get specific infections, whether it's Zika, and do well, whereas others may have a bad outcome. And so the current thinking really is that a host's response to a viral or any kind of infection is, of course, reflective of their the strength of their immune system, which is simplistic, but is really the bottom line. So what we look for in using these panels and the exomes and our approach are the genetic causes. Why is it that person A can get Zika virus and make it through all right, and person B may not make it at all and end up dead? And so it's a complex question, I think not specific just to Zika, but the answer being is that, of those 10,000 genes, there will be a constellation of a few, as we all have many mutations, that predispose some to having more or worse viral infections because perhaps their T cell function isn't as strong.
Okay, and Dr. Chou, this next one is for you too. What was the variant yield of the targeted panels of the first screen versus outsourcing whole exome sequencing?
So the yield for the first screen was about 55%. And then that's using outsourcing whole exome sequencing. And then using the targeted panel is much higher. It's about 80%. And so the difference in that maybe the sample sizes aren't the same yet because, of course, we've just been doing the targeted sequencing for about a month. But that being said, if that holds true, the difference, I think that part of it's because we're able to get much greater depth of coverage using targeted sequencing than whole exome just because it's much cheaper. And we're able to much more easily pick up large exonic deletions. The copy number analysis of exomes is standard, but it's time-consuming and sometimes tricky and requires a bit of finesse. For the targeted panel, it's extremely easy to analyze and to identify these exonic deletions, and we've certainly found some study subjects who have had that as a cause of their phenotype. So I'd say that the targeted panel itself has certain advantages that we've found over exomes, certainly as a screening step.
Okay, and it looks like we have time for one more question, and it is, where can I get information about the AmpliSeq Primary Immune Deficiency Panel? Okay, you can go to ampliseq.com and search for Primary Immune Deficiency to get panel details. I would like to once again thank Dr. Janet Chou and Wayne Bainter for their presentations. Do you have any final comments?
No, it was a pleasure to be a part of this. Thank you very much.
Thank you very much for the opportunity.
I'd like to thank the audience for joining us today and for their interesting questions. If we did not get to your question today, the sponsor and the speakers will try to address them after the presentation. And we would like to thank our sponsors, Thermo Fisher Scientific, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through December 22nd, 2016. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.